Assalamu alaikum. Coming to another topic, the name of that topic is the structure of hemoglobin. Structure of hemoglobin, as we have discussed earlier, hemoglobin is divided into two. Hemoglobin, as the name, this portion of heme, and this portion is globin. Heme is further classified into two portions: iron, that is ferrous form, plus porphyrin. And we have already discussed that how this porphyrin is manufactured inside a cell. When we have drawn some great doctors prescribe that sentence. Some great doctors prescribe highly cancer specific pills. So these were the pneumonics globin. Now we are discussing the structure of hemoglobin in detail, so we will come to know about this, why there are certain pathologies which are related with the human body, like thalassemia, like sickle cell anemia. So for that we have to discuss this normal structure of this whole thing. So first of all, coming to the structure of heme. Heme has a structure when there is iron in the middle. This iron is in ferrous state. There are 4 pyro rings here. This pyro ring number 1, this pyro ring number 2, pyro ring number 3, pyro ring number 4. So there are 4 pyro rings. We count it like 1, 2, 3 and 4. So their total pyro rings are 4 in number. These pyro rings, on every angle, on every spot, they have CH bond. So we write CH, CH. On this end, it has nitrogen. So CH, CH, CH. And this nitrogen in between. These all pyro rings are attached with one another with the help of certain bridges. These are that bridges. So for differentiation we wrote 1, 2, 3 and 4. These bridges are called methylidine bridges or methine bridges. Now coming to the bonding, there are double bond present here, double bond present here, double bond present here, double bond present here, a double bond here, a double bond here, double bond here and a double bond here. This iron, which is in ferrous state, it is likely to attach with 6 other atoms. How it attach with 6 other atoms? Like in this valencies, H4, Fe, CN6. So this iron is attaching itself with 6 atoms. So how it complete its valency here? It attaches itself with one nitrogen. It come on attaching itself with another nitrogen. This nitrogen is attaching itself with this iron molecule, and this nitrogen is attaching itself with this nitrogen molecule. So to summarize it again, there are 4 pyro rings. These 4 pyro rings are attached with one another with the help of methylidine bridges, methylidine bridge, methylidine bridge, methylidine bridge and methylidine bridge. To separate these numbers we have wrote 1, 2, 3 and 4. And to separate these methylidine bridges from one another, we have also classified this in alpha, beta, gamma and delta. So there are four numericals, or you can call it A, B, C, D, or 1, 2, 3, 4, whatever you want. So that this can be differentiated from A, B, C, and D. So these four Haemoglobin bridges can be separated from one another on this basis as well. Coming to again this iron, which is in ferrous state, it has attached itself with 4 nitrogen molecules, one from each of the pyrrole ring. It attached itself with another. This heme portion, this hole is called heme. This hole is called heme. This heme portion is attached with globin portion with the help of this iron molecule. So this is its fifth valencies here. Where in globin chain this iron is attaching? In globin chain there is amino acid with the name histidine. This iron is attaching itself with the molecule with the histidine amino acid. Histidine amino acid is a bigger amino acid. Where in histidine amino acid? So the nitrogen molecule in histidine, it attach itself to that nitrogen molecule. So iron is attaching itself with 4 nitrogen molecules here in the 4 pyrrole rings. It attached itself with 5th nitrogen. And where is this 5th nitrogen? This nitrogen is present in the molecule of histidine. Where is this molecule of histidine present? It is incorporated in the molecule of globin. So this is the 5th valency of this iron. Coming to the 6th valency, it attached itself with the molecule of oxygen. Whenever the conditions are oxygenated, it attaches itself with the molecule of oxygen, and whenever the conditions are not oxygenated, the conditions are deoxygenated, it attaches itself to the molecule of water. So this was the basic structure of heme, which we were describing. So for again, let me summarize it. There are 4 pyrrole rings. Pyrrole ring number 1, pyrrole ring number 2, pyrrole ring number 3, pyrrole ring number 4. These pyrrole rings are attached with one another with the help of methylamine bridges. In between there is an iron. This iron is present in the ferrous state. This nitrogen molecule is attaching itself with iron. So iron is attached with 4 nitrogen molecules. It is attached with another nitrogen molecule. This nitrogen molecule is a part of histidine, and this histidine is a part of globin. So this is heme, and this is globin. This iron molecule can bond with oxygen molecule when the conditions are oxygenated, and it can attach itself with the water molecule whenever the conditions are deoxygenated. One hemoglobin molecule has a tendency to combine with 4 oxygen molecules. So remember, one hemoglobin molecule can attach itself with 4 different oxygen molecules. These methylamine bridges are also numbered alpha, beta, gamma, and delta. So I just remove it so that we can view it more clearly. On the outer surface, this is 1, this is 2, this is 3, this is 4, this is 5, 6, 7, and 8. If this iron is plus 2, it is converted itself and it get an extra charge. If it is plus 3, it means it get an extra charge. To overcome this extra positive surplus charge, it has to be attached with a negative molecule, which is most likely Cl negative, or SO4 negative molecule. In certain conditions, what I want to say that in certain conditions, this heme, this iron portion contains a surplus charge. And if it contains a positive surplus charge, so to overcome this surplus charge, it has to attach itself with another molecule which contains a negative charge. So when it get attached to a negative charge, this one positive charge and this one negative charge, these cancel the effect of one another and no significance pathology will be formed. Now coming to these numbers 1, 2, 3, 4, 5, 6, 7, 8. So these are those 8 carbon atoms which are not part of methylidene bridges. So carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6, carbon number 7 and carbon number 8. These 8 carbon molecules are not part of methylidene bridges. There are a poison with name cyanide. If a person takes cyanide poisoning, what this cyanide poisoning do? There is a hormone, this is an enzyme with the name cytochrome oxidase, which is present inside mitochondria and it is responsible for electron transport chain. So cyanide, what happens when a patient takes cyanide, it affects this cytochrome oxidase and it blocks this cytochrome oxidase to be effective. So how you treat this patient? You give the patient NaNO2. You give the patient NaNO2 or sodium thiosulfate. What this sodium thiosulfate or NaNO2 is doing in the body? This sodium thiosulfate or NaNO2 converting hemoglobin to methemoglobin. It converts it into methemoglobin. Methemoglobin is the favorite site of this cyanide to be attached. So methemoglobin goes and attaches itself to the cyanide and what it forms? It forms cyanomethemoglobin. So this cyanide is going and attaching itself with the methemoglobin and what you get? Cyanomethemoglobin, and as a result this cytochrome oxidase will be not under the effect of this cyanide poisoning. And if this cytochrome oxidase will be not in the effect of cyanide poisoning, so this mitochondria and this electron transport chain will be effective and it will perform its action. Let me recap this thing. If there is a patient with cyanide poisoning, what happens? There is an enzyme with the name cytochrome oxidase which is present inside the mitochondria and it is stopping this electron transport chain. So what you do? You give the patient NaNO2 and sodium thiosulfate. NaNO2 or sodium thiosulfate, what is doing it? It is going to the hemoglobin and converting the molecule of hemoglobin into methemoglobin. What is methemoglobin? The methemoglobin is stopping the effect of the cyanide. How this methemoglobin is getting itself attached to cyanide and you get cyanomethemoglobin, and as a result this cyanide cannot act on this. Instead this cyanide get attached itself to methemoglobin and what you get? Cyanomethemoglobin molecule.